Hello, my name is Elizabeth Wright, and I am a former PhD student at the University of Arizona. Today I'm excited to show you some results from our most recent short report published earlier this summer, in which we uncovered a role for transmembrane protein 184B in endolysosomal acidification via the vesicular proton pump. Previous work identified a role for this 7-pass transmembrane protein in neuronal maintenance as in a gene-trapped mouse model, swollen nerve terminals and presynaptic membranous inclusions are observed. However, the mechanism by which TMEM184B regulates synaptic maintenance is still unclear. In this study, we first identified that TMEM184B interacts with proteins that are known to regulate intracellular transport and endosomal trafficking. We found specifically that TMEM184B interacts with proteins that regulate vesicular acidification, in addition to interactions with a specific subunit of the proton pump, otherwise known as the VATPase, suggesting a potential role for TMEM184B in endosomal trafficking through acidification of compartments. So to test if TMEM184B regulates endosomal acidification, we use the Firefly pH sensor developed by the Gao lab at UCSF. We specifically found that in the absence of TMEM184B, LAMP1 positive puncta are less acidic, supporting our hypothesis that TMEM184B promotes neuronal endolysosomal acidification. We next tested if TMEM184B promotes endolysosomal acidification through regulating VATPase functioning. Using co-immunoprecipitation to pull down the V0A1 subunit and its interacting V1 domain subunits, we assessed VATPase assembly. Here we identified a significant reduction of domain interactions in the absence of TMEM184B, providing an explanation for reduced puncta acidity we previously observed. Together, these results identify a mechanism by which TMEM184B regulates neuronal endolysosomal acidification to maintain synaptic structure and function. Future studies aim to better understand the functional role of this 7-pass transmembrane protein to better treat TMEM184B related neurodevelopmental disorders and neurodegenerative diseases characterized by disrupted endosomal acidification. Thank you so much for your attention, and I encourage you to review the rest of this short report online.